really delighted today to welcome Dr. Michael Friedman for our CEMB Future Leaders in Mechanobiology seminar. Dr. Friedman is currently a research assistant professor at Virginia Commonwealth University. He started his academic career at UT Austin, where he majored in biomedical engineering, and then went on to Michigan to carry out his PhD studies, working principally in the laboratory of David Cohen. In that role, he studied how exercise combines with calcium and phosphorus supplemented diets to impact bone remodeling and mechanics in mice. He then went on to a postdoc at VCU before taking on his faculty position, where he worked with Hank Donahue, studying how limb immobilization impacted bone remodeling and mechanics. And he's going to tell us today about some of the more recent work he's done studying genetic variability and how this impacts the response of the musculoskeletal system to disuse. Dr. Friedman has won multiple awards in his academic path. This includes grants as a postdoctoral fellow, a fellowship from NASA Translational Research Institute, and more recently, a NASA individual grant as PI. He's been honored as a rising star in various symposia, including at Columbia, Johns Hopkins, and Boston University, and won the Young Investigator Award at the Advances in Mineral Metabolism conference in 2020. He also in 2019 won the very prestigious Alice L.G. Young Investigator Award from the Orthopedic Research Society, where he plays active roles and in fact is a member of their outreach committee. So with that as introduction, Michael, we're really excited to have you here today with us and are looking forward to your presentation.
Thank you. Thanks for the introduction, and really happy to be here today to talk about my research. And first of all, I'm going to go ahead and give you some background information about me so you can get to know me better and where I came from. So I was born and raised in McAllen, Texas. So it's a town on the southern border of Texas, and my mom's from Mexico, and this region of the state is very not diverse. As you can see by the census, it's almost entirely Hispanic based on where we're located. And about this region, it's a metropolitan area, but it's the poorest area in the United States, and a quarter of the population is below the poverty line. And as a result of this, we have very poor healthcare, very low access to healthcare, leading to high obesity rates. And so a lot of this growing up in this region has influenced a lot of what I do in science. And if you're wondering, their climate is just hot and hotter. So also a big thing, since I'm from Texas, football is extremely important to me and my family, and that's my brother's a football coach. And so this got me interested in science and orthopedics research. I've injured almost every joint in my body, so I'm very interested in orthopedic technologies and ways we can have better care. So they mentioned my background and my education from the University of Texas and grad school in Michigan. And there, my research was in combining exercise with calcium supplements. So I was looking at how can we maximize bone strength, and what we found, I had just gotten started in bone research. I didn't really realize the significance of our findings at the time, but the longer I've been in the field, I realized how important it is. So typically in mice, these are black 6 mice, they have decreased bone volume over time with aging. And there's this one graph here that summarizes what we found very nicely. If you go from baseline and control mice that don't exercise, they have decreased bone volume over a month. And these are in adult mice. And then when you exercise, the prevents bone loss. So you don't have decreases with aging. But when we give just a simple mineral supplemented diet, no fancy interventions, and we see increased and sustained increases in bone volume. So this got me interested in how we can affect, or interventions to affect bone metabolism, and what can be done to prevent bone loss. And so in my postdoctoral work, I have a postdoctoral fellowship that's funded through NASA, and we're looking at different models on Earth to model the bone and muscle loss that occurs with spaceflight and disuse. And what was particularly of interest is what role does genetic variation play in bone loss, since all the astronauts in space are genetically different, whereas most research in the lab is with genetically unique inbred mice. So I'll go into more about this project now. And we know that spaceflight causes extensive bone and muscle loss. So bone loss can be about 5% per month, and muscle is much worse. You can have the 20% decreases in muscle volume per month. And we know through genetics that some people have higher bone mass and some people have higher muscle mass and muscle strength. But we don't know if genetics affects the response to spaceflight or the response to disuse. So maybe some people will be more protected from the catabolic effects of spaceflight. So typical studies in the lab use inbred mice where they're all genetically identical, or in vitro, we use cell lines that come from one mouse or one person, and all our cells are genetically identical. So this is similar to performing a clinical trial on just one person, and that's not realistic for how clinical trials are performed. So a better model in mice that would be more translatable would be if we use genetically unique outbred mice. This way we could account for the genetic diversity that is seen in the human population. And we know that many interventions developed in animal research don't translate successfully to people. So perhaps by accounting for the genetic diversity, this can help improve the translatability of models. So just to go over the differences between inbred and outbred mice, the inbred mice, they're all genetically identical. And this typically requires a low sample size to see effects of intervention. So in our lab, if we apply a disuse intervention to a mice, we can see significant bone loss with just 3 or 4 mice per group. However, this has limited power to test for effects of genetic variation. Whereas the outbred mice, they're all genetically unique. Every mouse that's born is different, and as time goes on, the offspring of those mice will be genetically different, and you can never have 2 mice that are the same. And this leads to huge variance in every phenotypic property, leading to a high sample size needed. So people are familiar with inbred mice. They do, even though they're genetically identical, they do all have somewhat different personalities, and they have somewhat different behavior, leading to variance in like body mass or bone mass. However, outbred mice, the range of personalities and range of behaviors that the mice can have is much broader, and you therefore have much higher variance in properties. So the Jackson Laboratory developed this diversity outbred mouse model to study the effects of genetic variation. And to do this, they took these 8 inbred mouse strains. Some of them are commonly used in research, like the Black 6, and some of them are more like wild mice and are rarely used, like the PWK and WSB strain. And to develop the outbred mouse model, they crossbred each of these strains many times, and after many generations of breeding, they led to this JDO strain of mice that are all genetically unique. And in our lab, we wanted to study the effect of disuse or immobilization on these mice to see how genetic variation affects the response to immobilization. And so as a first step, we looked at how do each of these individual inbred strains respond to immobilization to see if the outbred mice would be a good model to use. So in the first study, we took 4-month-old male mice from each of these 8 strains, and they all had one leg, the left leg, put in a cast for 3 weeks. And then this shows the % difference between the immobilized leg versus the contralateral untreated leg from the same mouse. And looking at the femur bone volume, you can see that the Black 6 strain had a very high response to immobilization with bone loss around 30%. And if you look at all the other strains, there was a wide variety of responses where about half the strains had huge bone loss and the other half had moderate to little bone loss. So this showed that because there was variance in the response from the founder strains of the outbred mice would be a good model for studying the effects of immobilization. So our main hypothesis is that genetic variability influences the musculoskeletal response to unloading. And this means that some mice will have genes that are protective against the catabolic effects of unloading, as well as some mice will have genes that amplify the catabolic effects of unloading. So in this study, we took 16-week-old DO mice. So this is the same age, the same intervention, and same duration that we tested in the founder strains. And here we put the right hind limb in a cast for 3 weeks, and then the mice were sacrificed at 19 weeks old. And all of these mice were single housed with unlimited access to food and water. They also had several enrichment items in their cage. And then at the end of the study, we evaluated the mice for bone and muscle properties. So overall, there were about 60 mice used in this study. And they're divided into 3 different cohorts, just to say that we didn't perform this immobilization on all the mice at the same time. We had 3 different groups, and the data I present will be combining all the data from all the groups of whatever we have available now. And so it's important to note this first group we had 20 mice, 10 female, 10 male, and this was the generation 45 of the DO mice. And then the other 2 cohorts were generation 46. In the first cohort, we had all the bone and muscle phenotype analysis complete, whereas it's still ongoing in the second and third cohorts. And then in the future, we will do transcriptomic and genomic analyses. So this shows you the body weight of the mice. So we received the mice at 6 weeks old, and as expected, the male mice generally had higher body weight than the female mice. The female mice were more along what you would expect to see in Black 6 mice, whereas the male mice had more propensity for obesity and generally higher body weight than what we would see in Black 6 mice. And then as you can see here at 16 weeks old, we started the mobilization intervention, and this caused a slight body weight decrease in both the male and female mice. And so this is important because a lot of the properties we're evaluating, like bone mass and muscle mass, are highly correlated with body weight. So if overall the mouse is having decreased body weight, we expect to see decreased bone mass and muscle mass in both the immobilized limb as well as the control limb. So first we looked at the femur cortical bone in the mid-diaphysis. So this data shows the in vivo CT scan on day 0 for all the mice. And we looked at the cross-sectional area and looked at the cortical area over the total area. So that's the cortical area is the white bone part, and the total area is the bone plus the marrow. And here's some examples of our mice where one has very high cortical area fraction, and this is more of the average. And so we saw here, there's extremely high variance, especially in the female mice. So the range is much more than you could have in any of the inbred founder strains, and much higher differences than we typically see in transgenic mouse studies that affect these bone properties. Another interesting thing that we observed was for all these mice, the right leg was significantly higher cortical area fraction than the left leg. And this was not well documented in the literature and not well seen in some previous work done. You can see here, this shows that limb was significant, but the percent of variation from individual mice is very high at 90%. Typically, what we've seen in black 6 mice would be around 70%. So we're getting huge variance just from the start, and it could have a big effect in response to mobilization. So here, after 3 weeks, we showed, we took an in vivo CT scan at the end of the study, and this is the percent change from baseline CT scans taken on the first day. And so you see that both the female and male mice had significant decrease in cortical bone. So you see in the control leg, there's a wide variety of responses. Some mice actually had a large gain in cortical area. Some mice lost cortical area in the control leg, whereas most of the female mice lost cortical area in the immobilized leg. And you can see here that the magnitude of loss seemed to be higher in the female mice than the male mice. And this was a consistent trend that we saw where the female mice seemed to be more affected than the male mice. And next, we looked at trabecular bone, in particular in the distal femur epiphysis. So this is a three-dimensional metric of the trabecular bone structure. And we're particularly interested in the epiphysis because this region has been shown in all of our previous work to be most affected by disuse. And we take these two-dimensional slices and analyze them. And you can see here a variety of looking at a mouse with very high trabecular bone volume fraction versus one that's more average. And looking at the data at baseline, we have this huge range between 20 to 60% VVTV in the female mice, and the male mice also had a high variability in this region, but not as much. And so we also surprisingly found that the female mice had significantly more bone volume than the male mice. And then here, the variation again was very high, not as high of a variation from mice as the cortical bone, but this is still much higher percent variation from the mice than you see in inbred strains. And looking at how mobilization affect the bone volume, we see a wide variety of responses. Here, just some pictures from this one mouse that had high trabecular bone volume at the beginning and had very little effect of immobilization, whereas this other mouse that had about an average amount of trabecular bone volume had a very big decrease from the immobilization. And similar to cortical bone, the female mice had more decreases in trabecular bone volume than the male mice. The male mice had this huge variability in how much bone volume could change. So we'll have to look into that in the future to investigate what's going on. The percent of variation from mouse was also very high for this type of intervention. We expect all the immobilized legs to have decreased bone volume, but we don't know how genetics will affect the magnitude of bone volume loss. And so to look at how variance in this response to immobilization compares to the commonly used black 6 mice, I looked at the coefficient of variation. So this is the standard deviation divided by the average, and using just the properties that are the percent change from baseline. So you see here in a previous study in our lab in black 6 male mice of the same age, the same duration, same intervention as we use in outbred mice. And you can see the coefficient of variation for various cortical and epiphyseal bone properties. And then when we looked at the DO mice, we found, and sometimes as much as 40 to 50 times the variance that was seen in the inbred black 6 mice. And we saw higher variance in the males. That was evident when you looked at the graphs of the data, but the female mice still had higher variance than what we saw in black 6 mice. So next we looked at the femur strength by doing this 3-point bending test to failure. So looking at the ultimate load of the femur, we saw a significant decrease in bone strength, but only in the female mice. So it wasn't like the male mice had a decrease, but wasn't significant. It was very striking difference. All of our other disuse models, we've seen a significant decrease in bone strength. So I'm not really sure why it didn't happen here for the male mice. You see, again, the mouse has a high percent of variance in this data, just like all the other properties. And we'd have significant sex and limb interactions for both ultimate load and ultimate stress. So the female mice had generally more bone loss in the cortical bone and trabecular bone. And then when we looked at bone strength, that translated to only the female mice having significant loss of bone strength. And then the last thing we looked at was muscle properties. So just the gastrocnemius mass that was weighed after dissecting from the mice, we saw significant decrease in muscle mass in the immobilized leg for both the female and the male mice. And prior to sacrifice, we tested the muscle strength in both legs of the mice immediately before they were sacrificed. And this shows the maximum tetanus force, which would be the maximum strength that the leg can produce. And we saw no significant differences between the control leg and the immobilized leg. However, we don't have baseline data, so it's possible that at baseline, the right leg was much stronger, and then after we immobilized it, it decreased such that both left and right legs were equal. And then looking at the twitch force, we saw, and so this is the maximum force produced by a muscle twitch. We saw increase with immobilization in both the female and male mice. And so this indicates that the immobilization is changing the function of the muscles of the immobilized leg. So overall, we saw that at baseline, the right leg had greater femur cortical area than the left leg. And so we were immobilizing the leg that had more bone mass to begin with. And we saw that immobilization decreased cortical area and trabecular bone volume, with generally a greater magnitude of bone loss in the female mice, and therefore only the female mice had decreased bone strength. The variance in how much bone loss and how much muscle loss occurred was much higher than what we would see in Black 6 mice, and immobilization decreased muscle mass and increased twitch force, indicating a change in muscle function. And a lot of these changes in muscle mass and bone mass importantly occur generally in most of the mice, so sort of independent of genetic factors, we're still able to see bone loss from this model. Where the genetics come into play is determining the magnitude of bone loss and what causes some mice to have more or less than the others. So future work on this project will need to look at which genes are affecting bone and muscle loss. We're going to do RNA analysis of the gastrocnemius and tibia, as well as all the mice are currently being genotyped, so we can see what genetic differences occur in them. And then in the future, in my future lab, I like to use outbred mice to test therapies for strengthening bone to prevent fractures, and look at how bone interacts with other tissues like skeletal muscle, the digestive system for nutrition, and look at kidneys or parathyroid gland for calcium metabolism, using outbred mice and primary cells from them. And so with that, I'd like to thank my lab and all of our funding sources. We have funding from NASA, NIH, and this was funded by a pilot grant from the Jackson Laboratory. And thank you, and I'll take any questions now.
Outstanding. Really interesting talk, Michael. So for questions, if folks want to add a question to the chat or just raise your hand on the Zoom, we can call on you one by one. Maybe while people are gathering their thoughts, oh, there's already one. Maybe I'll just ask the first question and just give a comment. So Michael, that was really a great talk. And I just want to commend you on this really important work that you're doing. I always joke with my students and trainees that, you know, I study OA and cartilage wear. I always make the joke that, we've cured arthritis in mice 1,000 times and in rabbits 100 times, but in humans never, right? And I suspect that a lot of that comes from the model systems we're all using that might predispose us to finding an effect size when really in an outbred strain, that effect size is much harder to see. So I think the work you're doing is just super important for teaching the field how to do science well. My science question is, I think the way you guys did this using in vivo CT so that you could look at indicial bone mass and morphology and function, and then follow that out after the immobilization is really critical. And I guess I was wondering, at the beginning, you showed us this really nice graph of the different inbred lines and how some of them show really massive loss of bone and others really hardly at all. Does the data that you have now allow you to build hypotheses about how much bone you start with relative to your probability of losing a lot of bone? So like, does this data set identify thresholds for disuse where if you're above that, you're probably good, but if you're below that, then you're going to have a really severe outcome?
Yes, thank you. That's a great question. And I believe so. Like I said, I completed the analyses for the first cohort, so the first third of the mice, and for the other two, it's ongoing. So some of these more in-depth analyses where you look at what properties are correlated to something, I'm sort of waiting until we get the full data set to dive into that. But yes, there's definitely the initial bone is going to be strong. Initial bone area or bone volume is going to be strongly correlated to the final bone area, regardless of what interventions we put in the mice. So it's really important to have those time series measurements so we can compare changes. And like we hadn't extensively looked at baseline metrics before to see that the left and right leg were significantly different. So knowing that the right leg starts off with more bone mass, if we only looked at the final day and we saw no difference between the right and left leg, we might think that nothing happened when clearly it would show bone loss in just the right leg.
Yeah, that's great. I can't wait till you get through all of this outstanding data. So
yeah. Oh yeah, and just as a follow-up, yeah, unfortunately we haven't, we're mostly a bone lab, so we haven't thought of doing extensive muscle analyses. And I think in the future it would be good to have baseline muscle strength metrics, because I think that's what was going on, that we saw no difference in muscle strength in the immobilized leg. But maybe if we knew what it was to begin with, that would help us better understand what's going on.
Great, thank you. All right, I think there's a question in the chat from Alec. Do you want to unmute yourself and ask your question? Or I can do it for you if you'd like. All right, Alec's mic isn't working. So on behalf of Alec B, I'll ask, with the increase in variance and differences in variance between the females and males, how do you determine the necessary sample size for studies with outbred mice? This is a question on everybody's mind, yes.
Yes, so for us, we predicted based on previous data using the inbred founder strains. So we do have some genetic variance in our past data to look through, but if you started with just black 6 mice, which most people do, it might be a little bit more complicated. And so based on the effect size we saw in our previous studies, we could predict how many mice we needed. And the answer of how many mice do you want, there's really no limit. It's sort of like people. if you're doing a clinical trial, how many subjects do you want? Ideally, if you had infinite resources, you want to test everyone. So we want to test as many mice as possible. And just using our previous data, we can predict maybe the minimum number we would expect to need to see significant effects. But like that paper we did with the inbred founder strains, that was before we had in vivo CT. So it's very clear here we could get significant results with 8 to 10 mice because of how powerful that tool is for us.
Excellent, thank you. All right, other questions? I have a couple more if no one else does.
I had a question.
Okay, please, Medora, go for it.
That was a great talk, Michael. Thank you. From your data about the differences, the baseline differences between the right and the left limb, could you elaborate a little more on that? Like, why do you think that happens?
Yeah, so I don't know why specifically. I think it would be extremely helpful if we had baseline muscle data to see if maybe one leg is stronger, and like, are these, because this is a new strain of mice that Jack's developed, so they haven't been fully characterized. Are they right leg dominant versus left leg dominant? Maybe some extensive gait analyses. Do they prefer to start off on the right foot versus the left foot? Do they sleep a certain way, putting more weight on that leg? There's a lot of potential to follow up on that. What we've seen is in the founder strains, we see a similar effect where the right leg is has greater bone mass than the left leg, but it's been very age dependent. And so this is only done in 4-month mice. And so it's possible that this effect doesn't exist in older mice or during development either.
That's very interesting. Thank you.
All right, maybe I'll ask, oh, Matt has
a question too. Hey, Michael, it's a great talk. So we did a little bit of studies with these DO mice when I was at UConn with David Rowe during my postdoc. And back then, this is like 8 to 10 years ago, it was like prohibitively expensive to use these mice. And oftentimes you would design simpler studies and then, divvy out tissues to multiple investigators to help save on cost. So can you speak a little bit more about using these mice now currently? Is this something that you're funding independently by yourself? Or are you going in, sharing costs with other labs? And just a little bit more of the actual logistics of getting the mice.
Okay, yes, it's a great question. So I believe the current rate on Jack's is around $80 for a weanlings. So it's not as expensive as maybe some of the rare strains that they have. And for us, the cost wasn't an issue because the grant we have from Jack's paid for the mice, and they currently have applications available now for grants where you can get free mice. As far as dividing up the cost, so because there was no cost, but we did have other tissue samples that were sent to other labs, and they're doing their own research on them. Other musculoskeletal research labs in our department. It's hard to give the tissues to people that aren't maybe interested in how immobilization would affect whatever tissue they're looking at. Like cardiovascular function might not be super exciting, but other people doing orthopedics research were interested in working with us.
Yeah, I would say Rob and I are both interested.
Yeah,
we had extra tissues.
Yeah. I was wondering if Nat was going to ask you for the Achilles. From
Yeah. That was my follow-up question. Yeah.
Yeah. I guess my last question would be, so you guys did the immobilization that you did is severe, but not injurious, not to the level of, a catastrophic, a failure event, significant overload. I know this is just idle speculation, but based on your experience with these animals, is everything we know about fracture repair also going to be completely reset by this strain of animals? What are your thoughts on that sort of thing?
I don't believe it's going to be reset. I think we saw similar to what you see in black 6 mice. There's bone loss with immobilization. It's just there's a variance in the magnitude. So I think the change in like callus formation will occur the same in these mice. It'll just maybe change how long it occurs, it takes for healing to occur, or the volume at a specific time point will have more variance in these mice than you see in normally with black 6 mice. So it's not, I mean, there's still mice, there's still interventions that apply to inbred mice. The black 6 are founder strains. So these mice did descend from black 6. They have black 6 genes in them. So it's not going to be hugely different. You're just getting much more variance in the response. And for now, I mean, usually they backcross inbred strains to keep prevent the genetic drift. These mice, that doesn't occur. So they could be evolving and change over time. We don't know. That's another exciting part of working with these mice. Every study we do is like a more advanced generation than the previous studies. And so they could be changing a lot over time.
Mhm.
Is it okay if I ask a question?
Sure thing, Hank, go for it.
So this demonstrates how independent Michael has been in this project, because I didn't know of these data. But Michael, how come the twitch force increases in disuse?
Yeah, so that has been a big question that we don't know the answer to and have to investigate. There's different studies in the literature where, depending on which muscle is immobilized, you could have increased or decreased twitch force. And I think with this, with the leg, you would expect decrease. So we don't know exactly what's going on now, and that's what we have to do further investigation.
Could it be that some muscle groups are compensating for the disuse of other muscle groups?
It could be.
Neither of us are muscle biologists, so we can say whatever we want.
Outstanding. All right. I saw someone turn their camera on. Was there one more question out there? It came on and then disappeared, so perhaps not. All right, well, not hearing any others. Again, Michael, thank you so much, not just for an outstanding presentation, but also for this like critically important work you're doing for the field. I know how much work goes into it, so just very appreciative of the work you're doing and its impact on our field. So let's thank Michael again.
Thank you.